Hello, my name is Vasundhara Agrawal, and I'm the Applications Manager for Spatial Bioimage Analysis at Leica Microsystems. Today, I would like to go over our collaborative effort with cell signaling technology to investigate the spatial architecture of tumor and immune cell lineages in syngeneic mouse tumor tissues. We know that immunotherapies have emerged as promising treatments for advanced cancers. However, immunotherapies may show limited efficacy, and this is because the optimal therapy for a patient is associated with the complex immunosuppressive mechanisms, as well as the tumor infiltrating immune cells within the tumor microenvironment, or the TME. Therefore, to understand the mechanisms of immunosuppression, we utilized FFPE tissue sections from syngeneic murine cancer models and performed tumor immune profiling to spatially understand the contributions of diverse cell types. Specifically, in this study, we utilized cell-dive multiplex imaging solution, which allows probing and imaging of dozens of biomarkers on a whole single tissue section with an iterative staining and dye inactivation workflow. Additionally, cell signaling technology has a broad portfolio of validated antibodies to detect key proteins in the TME, enabling tumor and immune cell detection. So let's take a look at Table 1, which highlights the panel of CST immuno-oncology antibodies used in this study. Tissue sections were iteratively stained with this panel of antibodies, and we applied this panel in rounds of 4 biomarkers using the cell-dive workflow to 3 syngeneic murine cancer models. The cell-dive multiplex images were then used for segmentation, followed by downstream spatial analysis using AVIA software. Here, we demonstrate multiplex cell-dive imaging using CST antibodies across these 3 mouse syngeneic tumor types, as shown here. We can appreciate how cell-dive can allow us to visualize spatially distinct heterogeneous regions, especially marked by metabolic markers such as GLUT1, which is a marker for tumor hypoxia and is shown in cyan in these images. Walking through Figure 2, in Panel A, we demonstrate clustering by expression levels of tumor hypoxia marker, GLUT1, to show distinct immune cell types enriched within CT26 WT syngeneic mouse tumor model. Panel B shows 3 major clusters that were determined based on the level of expression of GLUT1 biomarker within individual CD45 positive immune cells. The key takeaway of this analysis is that overall, we find that clustering by GLUT1 expression levels reveal differential enrichment of immune cell types. Specifically, GLUT1-rich regions were enriched in myeloid cell types such as CD11b, CD206, CD11c, along with some immune checkpoint expression. Also, the expression of markers associated with mesenchymal cells, such as vimentin, and immune evasion, such as CD47, may indicate tumor adaptation and resistance developed within GLUT1-rich regions. Also, increased infiltration of myeloid cells, as well as immune effector molecules such as iNOS and granzyme B, suggests an active immune response within these GLUT1 positive regions. At the same time, within the GLUT1 poor regions, we note enrichment of T cell markers such as CD3, CD8, as well as a marker for regulatory T cell activation, IL2RA, along with some immune checkpoint protein expression. Therefore, this analysis highlights the spatial expression profile of tumor hypoxia along with immunosuppressive populations. But this makes us think about how all other immune markers are expressed within the whole tissue. To answer this, in Figure 3, using unsupervised clustering on AVIA analysis software, we can identify various immune cell populations enriched in tumor cells within A20 tissue in panel A, and CT26 WT syngeneic mouse tumor model in panel B. One such immune cluster is highlighted in green. In green here, we see T cell clusters showcasing immune checkpoint protein expression of CTLA4, TIM3, and PD1, along with markers for regulatory T cell expression, including FOXP3 and IL2RA, indicative of potential immunosuppressive mechanisms impacting T cell function in the tumors. Next, in pink, we see myeloid cell clusters showing M1 and M2-like functional markers for macrophages, like INOS, CD206, respectively. And PDL1 co-expression in these cell types further suggests a myeloid-based immunosuppressive mechanism. Also, we again note the enrichment of GLUT1 expression in these myeloid cell populations in both kinds of tissues, possibly aiding towards tumor immune evasion and resistance to therapy. Overall, such clustering-based analysis helps us identify immune cell types and subtypes within the tumor microenvironment. In conclusion, cell type multiplexing of the CST biomarker panel combined with AI-powered image analysis using AVIA allowed the study of immune landscape and molecular characteristics as delineated by GLUT1 expression, which is intricately linked with metabolic reprogramming and regulation of immune activation within the TME. Such studies could provide mechanistic insights into the tumor microenvironment, which will help strategize new therapeutic combinations targeting metabolic reprogramming along with immune pathways. Finally, if you're interested in taking a closer look, the poster is available for download on our website. Thank you for listening.